Very excited to have Dr. Chelsea Cook visiting us today. So Chelsea is visiting us from from Wisconsin. She is currently an assistant professor at Marquette University, hired there in 2020, so brand new prof. Prior to that, she was at Arizona State University doing a postdoc, and she did her PhD at the University of Colorado Boulder with Mike Breed. So you're going to see some really cool bee work today. And I think I forgot one other thing. She's also a startup founder. So Dr. Cook runs Hive Tech Solutions. It's kind of cool. Looks at honeybee cold storage. So you can ask her about that as well at the end.
Absolutely.
So Chelsea, I'll let you kick it off. I'll mute myself and thank you.
Awesome. Yeah. Thank you all so much for coming. I'm so excited. I know, beginning of the semester is kind of tough to motivate to get to extra things, especially as we all kind of navigate these weird times. So I really appreciate it. And thank you so much, Brock, for organizing and the invite. I'm really humbled to be able to give a talk today. So thank you all so much. So as Brock mentioned, most of my talk today will be about some of the work that I've done. I did as a postdoc, and coming from this, kind of perspective of behavioral ecology, individual and collective behavior, focusing on honeybees. So I have a little bit of gene expression stuff at the end of my talk. I had a lot of hopes to get more in here, but as the semester began, and a colleague of mine had a baby, we didn't get as much done as I wanted to. So there'll be a little bit of gene expression, but I promise there'll be more coming out for my lab. So that being said, thanks again, and we'll go ahead and get started. I do want to, of course, take a quick second to just say thank you so much to all of my supporters and collaborators. So I did my postdoc on a NIH postdoc fellowship. So I'm very thankful to them for that funding, which was really incredible. Really supportive labs. ASU is a great university to be at for a postdoc. And of course, none of this work gets done without amazing undergrads, high schoolers, graduate students who do this work and help really keep me going and help keep the research going. So I have an extensive list here of amazing students who helped me collect this data, and I will shout them out as I go through my talk as well, because they are incredible. So this talk is really going to focus on investigating collective behavior. So here I have several photos of some of the most remarkable collective behaviors that happen on this planet. So in the top left is army ants building a bridge, literally building a bridge with their bodies across a gap. The photo of the honeybees on a feeder is a photo that I took. So if you set out a feeder full of sugar water, within a couple of hours, these feeders just get absolutely bombarded by honeybees. So they find these feeders very quickly and are able to collect as much food as possible from them. Down on the bottom left, we have a school of barracuda who are schooling and performing this really incredible tornado behavior to school and potentially avoid predators. And then, of course, we have, the humans at the stock market, right? So this massive collective behavior, these individuals making these choices and making these decisions that drive a lot of and have a lot of influence over our society, especially in the human case. So and then in the honeybee case as well, right? So humans, depend on pollination and these behaviors from honeybees. So I take a look at these groups and I'm absolutely fascinated by the collective behavior, but I can't help but look at these groups and say, each of these individuals has their own genetic background, even though they may be closely related in the case of ants and bees. They have their individual genetic background, their individual development and environment that they developed in. They're individuals, right? So they perceive and interpret and understand their world in their very unique individual ways. So I can't help but look at these groups and ask, how do these individuals come together and affect this massive change together? And so that is really the crux of this talk today, is how does this variation in individual cognition, so how individuals are perceiving the world around them and acting on that perceived information, how does that variation shape these massive collective behaviors? And so to answer this question, I take a multi-level approach. So my whole research program through my academic career has been investigating these behaviors from multiple levels. So at first I take the individual, I look at what she's doing in the case of honeybees. So how do individuals behave? What is this one individual doing, even in the context of the collective, in the context of different environments? What is she doing, and how does she potentially learn or behave in her unique way? And then I ask those questions about the collective. So how does the collective behave in different environmental contexts or in different social contexts? And then I kind of take a perspective looking at the individual an individual in these groups from a mechanistic perspective. So what's going on? Mostly I focus on the brains of these individuals. What's happening that drives this individual variation that then scales up to the collective? So I'll talk about all these levels today. First, I want to focus in on the individual level. So what types of individual variation do we see in honeybees, and what do how can we measure and actually quantify this individual level behavior to then get to the collective behavior? So as I've already mentioned, my system that I focus on is honeybees. Honeybees are fantastic model organisms because they're really easy to keep. They're really easy to get. Their genome is mapped, so any genetic or genomic analysis you want to do is relatively easy. And aside from the stinging, they're pretty cute. So they're pretty fun to work on. They're very charismatic, and most people absolutely love honeybees. So they're really fun, really great system to work on. Honeybees also exhibit quite a few pretty complex cognitive behaviors. And one of the, so when I started my postdoc was in Brian Smith's lab, and he's more of a kind of learning neurobiologist. So he's interested in what's going on in the brains of animals as they learn, as they navigate their environment. So I naturally kind of got drawn into those questions as well. So while I was there, I learned about this behavior called latent inhibition. And latent inhibition comes from the psychology, kind of learning psychology world, mostly rooted in humans. So, it's kind of a little bit of a complex phrase for a relatively straightforward behavior. So latent inhibition is when animals or individuals are able to learn to ignore familiar or unimportant stimuli. So it's different than habituation, where the animals, maybe the neurons in the brains of the animals maybe just get desensitized to certain stimuli. This is a measurable learned response. So this latent inhibition is specifically about individuals learning to ignore in stimuli that they've been exposed to that doesn't seem to indicate any important information. So in humans, latent inhibition is actually associated with ADHD and schizophrenia. So individuals with low latent inhibition exhibit inability to ignore unimportant information. So you can imagine in a situation with someone who has ADHD, they can't ignore unimportant information. So their brain says, pay attention to that, even when that individual maybe shouldn't be paying attention to it. It's also in kind of an ecological animal behavior context. It's related to predator recognition. So for example, if you're a fish swimming in a pond and a leaf falls onto the pond, is that a leaf or is that a predator? Over time, you want to learn that that's a leaf and not a predator, and be able to identify those cues and stop responding to that information. I wouldn't be telling you all of this if it was not exhibited in honeybees. So honeybees also surprisingly exhibit this relatively complex behavior. And we have a pretty really interesting standard assay to measure this. The variation in latent inhibition that we see in honeybees is also heritable, which is important for some of the work I'll talk about in a little bit. So this variation we see, we can measure in queens and drones, and then the offspring of those individuals tend to exhibit the same LI, latent inhibition, as their parents. So I'll abbreviate latent inhibition as LI often throughout this talk because latent inhibition is a little bit of a long phrase. So bear with me with that abbreviation. So to think about this latent inhibition assay in the lab, what we do, we have this automated system where we put bees in harnesses, and then we expose these bees to an odor. So these are constituent floral odors. We just buy them from Sigma. We use octanone and hexanol most often. And if you're familiar with those chemicals. And so we have this automated system where puffs of air go through these scented odor tubes directed at the bee. We familiarize them with 40 trials of just a puff of odor. So they're getting this puff of odor, no other stimuli, no other information along with this puff of odor. And then we give them a little bit of a rest period. So this is 40 times every 5 minutes. So this is 3.5 hours, over 3.5 hours of exposure to this odor over and over and over again. Then we ask them to learn how much have they learned about that odor. So we then pair that odor with sucrose reward. So we use the proboscis extension reflex. So we puff the familiar odor at them, and then the last second of that exposure, we touch their antenna, we give them a droplet of sugar water, and we evaluate how well the bees learn that odor. And we compare this to a novel odor, so an odor that they have not been exposed to. So we pair this 4 times with a novel odor, 4 times with that familiarized odor. And so this is what the proboscis extension reflex looks like. This is a great video from the University of Buenos Aires. I did not take this video. I wish I did. No camera that I own takes this good a video. So this is a really excellent video. So the honeybee you'll see is strapped in. So she's in this harness. She's strapped in with tape. Her antennae are able to move. So the odor comes on, you touch the antenna with sugar water, you reward them to the proboscis as they're being exposed to the odor. Then you say, have you learned this? Turn the odor on, the bee sticks out her proboscis, and that is a yes from the bee. Yes, I have learned that this odor is associated with a food reward. So really simple assay, a really high throughput. So we're able to familiarize those bees and then ask them, can you learn this odor? And so what we end up seeing after this assay is, so if I take, these are forager honeybees. So if I take the forager honeybees coming back to the colony, we can characterize them as high or low latent inhibition. So what I'm showing you now on this graph is individuals that are high latent inhibition. So looking at the blue dotted line, that's the novel odor. So they learn the novel odor really well, but that odor that we familiarize them to, they have a really difficult time learning that odor. So they're like, this odor didn't mean anything like a half an hour ago, and now you're telling me it means something. What's going on? I have a hard time learning this. So these are individuals with high latent inhibition. We can compare this to individuals with low latent inhibition. So you can see these two curves here, these low LIBs. So again, we don't know what they are before. This is post hoc characterization, that they learn the novel odor and the familiar odor in the exact same way. There's no difference. So these bees are like, sure, that odor didn't mean anything a half an hour ago. Now it does. Awesome. I got this. So they learn this odor, these odors equally. So it was really interesting. We see this variation if we collect 100 foragers returning. We see this variation, and we see high latent inhibition bees about 25% of the time, and high latent inhibition bees 25% of the time, low latent inhibition bees maybe like 50% of the time. And there's this chunk of bees that we don't see, we can't really characterize. They're kind of in the middle and not at these extremes. So there's some obvious genetic components, something going on there, which I have some data for, definitely getting that out soon. So hopefully you'll see that. So when we think about this variation, we see these foragers come back and we're able to kind of distinguish these into two groups. As a behavioral ecologist, my natural question is why? Why do we see this variation? And so how does this variation translate into relevant bee world behavior? So when I joined Brian's lab, I immediately went to foraging. So why would you need variation in latent inhibition thinking about foraging? And we thought about this in the context of who's going out and what types of jobs are they doing when they're foraging. So in the honeybee colony, you have scout bees who are these foragers who go out and they're searching for novel information, and they end up ignoring familiar information. So they go out, they find a novel food source, they come back and they perform the waggle dance, which is what you see here in this GIF. So the scout bee performs the waggle dance. So she's saying, hey, this new food location is over here, giving all this information to these recruit bees who are following her around. So you can see these bees kind of following her as she spins, and they're getting all this information. She smells like that space. Sometimes they feed each other, and they're obviously giving this information about distance and angle from the sun with the waggle dance. So scout bees are saying, hey, this is a new spot. These recruit bees are, their job after they're recruited is to persist at that location and exploit it. So you have these scouts who are these explorers. They're always looking for novel. They're not very likely to go back to that same place. They're going to continue to search the environment for novel and ignore familiar. Whereas the recruits, they should tend to be persistent at familiar and continue to exploit the resource that they just found out about. So because scouts must learn to ignore known foraging locations, our hypothesis is that LI is linked to foraging behavior. And we predict that scouts, because they have to ignore familiar, and they can ignore familiar, that they would be more focused on novel and ignore familiar. So they're going to be high LI. Whereas the recruited bees will be, we'll call them unfocused. So, just in kind of, to keep it a little bit more, latent inhibition is a little, I don't know, a complex phrase. So we think about this as like they get focused, they focus in on the novel. For the high LI bees, the recruits maybe are unfocused, as in they can learn whatever information is given to them. They'll learn all the odors. They'll be more low LI. So to test this, we induce scouting and recruiting behavior by performing this pretty typical hive moving assay, where we move the colony out of the typical foraging range of a colony. And then on day one, we collected scouts. And so I did this by marking all the bees leaving the hive, and I collected them only when they had been gone for at least 10 minutes, so that they'd been at least searching the environment for a novel food source, because everything was novel. So I did this by marking them and changing the color of what I mark them every 10 minutes. So this is colored cornstarch on a paint roller. So this is a one-way exit, so the bees can leave. They get dusted with this colored chalk. Let me play this video again because it's kind of cool. So they get dusted with this colored chalk as they leave. And then this is temporary, so they'll end up cleaning this off of themselves throughout the day. But at least it stays on for about an hour, which is the duration that I need it to stay on. And then I change this every 10 minutes. So from 9 to 9:10, they're pink. From 9:10 to 9:20, they're blue, and from 9:20 to 9:30, they're yellow. So you can see here, it's a one-way exit. So I have the entrance of the colony sealed up and all the other areas sealed up with duct tape. So no bees can get in and give any foraging information to the bees inside. So these are the scout bees. They've left, they've searched the environment for 10 minutes. They come back, they cannot go inside. So I scoop them up, and we evaluate them for their latent inhibition. So we do this with scouts. When I'm done collecting scouts, I open up that colony and allow them to freely forage for 24 hours. And then the next day, at the same time, I come back and collect recruits. So Tom Seeley's work indicates that about 78%, give or take, of bees returning are recruit bees, of foragers returning are recruited bees. So we collected them randomly on day 2, assuming that most of the bees that we collected would be recruited bees. And Sydney was absolutely instrumental in getting this work done. She was out with me in the hot Arizona summer, collecting these bees in 110-degree heat. So she's a rock star. So definitely wanted to shout her out. So now I'm going to show you, so we brought these scout and recruit bees into the lab and we wanted to evaluate their LI score. So we did this with just a simple proportion of number of responses to the novel odor +1 divided by the number of responses to the familiar odor +1. So the higher the number, the more responses to novel, the fewer responses to familiar, those are high LI bees. The number closer to 1 are equal responses to familiar and novel. So those are low LIBs. So we characterize them as scouts and recruits. And sure enough, we did find that scout bees tend to exhibit high LI and those recruited bees tend to exhibit low LI. So this supported our hypothesis. So we're pretty excited. We're like, okay, so in the field, it seems like latent inhibition plays an important role in how bees are foraging. So we have a little bit of kind of an ecological context to this behavior. So that, of course, now we have this individual level variation and why this might be beneficial to the colony. So I wanted to then start to explore the collective. What's happening at the collective? So I mentioned earlier that latent inhibition is heritable. So we're able to select learning lines, and honeybees, another advantage to working with honeybees is that queens can actually be instrumentally inseminated. So you can collect drones, you can collect sperm from drones, and you can inseminate the queen with selected drones that you picked. So what we did was we tested queens and drones for their latent inhibition. So queens and drones, surprisingly, also exhibit latent inhibition. I know not a lot of work on drone cognition. Not a lot of people have focused on drone cognition, but we did find that we can test drones with this pretty complex cognitive behavior and get results, which is really cool. So we found high LI drones and high LI queens. We found low LI drones and low LI queens through these assays. And then we're able to do single drone inseminations with instrumental insemination with those queens and generate very closely related workers from those combinations. So what we did was we generated these pairings, then we waited for them to produce workers, and then we took the frames of brood from those colonies, threw them into incubators, waited for bees to emerge, and we marked them as they emerged. So on day 1, which is really nice because their bees are not hard enough to sting you, so you can grab them, scoop them up, dot them with the dot of paint. And we dotted them with the line of paint associated not only to their line, but to their colony as well. So we know which colony and which pairings these bees come from. So we're able to do this, and then we're able to put them into colonies, put them into the field, and ask questions about how they forage and how they learn. So first, we wanted to make sure that the social environment didn't influence the LI of the bees. So if we collected them later as foragers, we wanted to make sure that the social environment didn't influence the LI, didn't influence their LI. So here I'm going to show you these pairings. So these are across the x-axis are high LI bees that were put back into their own high LI colony. So we mark them, put them back, wait until they're foragers, and then collect them and test their LI. And then we took those same 50% of those bees we collected, we put them into a control colony, so a size matched colony that was not selected, so they have a variation of learning behaviors. We also did that with low Li bees, so we marked them, put them into a control colony, or we took half of them and put them back into their low Li natal colony. And then when they were foragers, we collected them and we tested them for their Li. So this is the same assay and the same calculation. So number of responses to familiar divided by number of responses to novel divided by number of responses to familiar. And we find that the high Li bees tend to stay high in their own colonies. The low Li bees tend to stay low in their colonies. And then when they are placed into a control colony with a variation of learning behaviors, they also continue to retain their Li. So social environment doesn't seem to affect the Li of the bees when they're foragers. Okay. So now we're getting into it. So we mark these bees, we keep track of where they're coming from, and then we can create colonies of these bees to put into the field and ask them questions about how they forage. So we create colonies of all high Li bees. So high Li bees, we tend to mark them warm colors. So you see pink, orange, red here. And then we have colonies of low Li bees. So you see cooler colors, green, blue, there is yellow, which is technically a warm color, but we use yellow for the low Li. And then we created a 50-50 mix. So you see a nice rainbow of colors here. So we want to see how these colonies of all high Li, all low Li, and a 50-50 mix forage out in the field. So we have these really wonderful hoop houses that are a controlled foraging environment. They're not enormous, but they're big enough to do the job. And they also will do round dances in these foraging arenas as well. So we get a little bit of that round dance information, which I'll talk about in a little bit. So we're out in the field. We place these colonies. So I don't know if you can see there's a small nucleus colony, so a 5-frame colony in between these tables. Water's available to them at all times because it is Arizona summer, it is hot and dry. And then we place different feeders. So feeders are different colors and different odors. So I'll tell you about those combinations in a sec right now. So we place a familiar feeder about 4 meters from the colony, and that familiar feeder is red. It has a specific odor, and that stays in the same spot through the entire week. So on day 1, they become familiarized to this one food location. And then on day 2, we introduce a novel feeder. So we introduce a novel feeder X. So this is a different color, different odor. On day 3, we introduce a second novel feeder. So this is feeder Y, different color, different odor from both the familiar and the previous feeder. And we do the same thing again on day 4. So for 3 days, the bees get a novel feeder to choose from compared to the familiar feeder. And I am doing this in 4 replicates. So I have a high colony, a low colony, a mixed colony, and a control colony that are age-matched bees from non-selected colonies. We did also have to supplement the selected colonies with control bees as well. If anyone has ever worked with single drone inseminated queens, you know they don't produce a ton of brood. So we combined lines, and we also supplemented with control bees just to get those numbers up. But they're all age matched, so they're all within a day or 2 of emergence from each other. So that's the experimental design. And then we're going out. So we have marked them on their thorax. We have waited 2 weeks of them being inside. So they're not exposed to the outside at all. They have never foraged before. We place them into these tents. And then as they visit feeders, we also mark their abdomens. So these 2 bees here, we have a high Li bee who has a pink mark on her thorax and a control bee who is unmarked. And they are visiting feeder X. And I know that because feeder X is the blue feeder, and we have marked them on their abdomen with a blue marker. So bees, another reason why they're great is when they're feeding, that is generally the only, if you do it gentle enough, is the only thing they care about. So you can dot them on their little abdomens and they don't really care. So it's really nice. So as they're feeding, we dot them on their abdomen so we can keep track of which feeders feeders they're going to. And we're out there collecting this data. So we're doing these amazing students, Alexa and Etta, were undergrads at ASU, and Nisha was a high school student who came out and learned like within a day how to collect these data. So she's such a rock star. She's an undergrad now at ASU. So we're doing scan sampling. So I have an undergrad out with me who's taking 2 tenths, I take 2 tenths, and every 5 minutes we pop back and forth, we mark bees, and we write down who is visiting what feeder. And so this is, I'm going to start showing you data. So this is just a characterization of overall visits to all feeders. I'm going to split this up by line. So this is just throughout the day, how are visitations happening at any feeder, separated out by line. So what we see is that the hyali bees tend to leave and visit feeders more often than the other colonies. The mix are kind of in the middle, and the low and controlled bees tend to make less visits, but they're all visiting the feeders quite often throughout the day, especially as the day progresses. So just wanted to get, brief characterization of what's going on in these colonies. I want to take a step back. So we're thinking about what would we actually see? What are our predictions? So we thought that the hyali colonies would prefer the novel feeders because they're able to ignore familiar. So if there's something that is equally as good, but it's novel, they may be more excited about it and they may be visiting those novel feeders more. So I forgot to mention that the quality of food in all these feeders are the exact same. So there's nothing different except for location, color, and odor. And the LowLate colonies, we hypothesized because they learn all the odors in the lab, that they would learn all the feeders and show no preference across the feeders. So what did we see? So here, now I'm splitting it up by colony. So we have high Li, mixed, low Li, and control colonies. And this is going to be split up by novel and familiar feeders. So I've combined all the visitation to the novel, and we have the visitation to just the familiar. So what did we see? So first I'll show you the results for the low Li and the control. So we found that as we predicted, there were no differences in how the bees visited these feeders. No difference, no preference in the low Li bees visiting familiar versus novel. So next I'm going to show you what happened in the high Li colonies. So I'll remind you, we predicted that they would prefer the novel feeders. They did not. They preferred the familiar feeder way more than they preferred the novel feeder. So those bees, once they found that familiar feeder, they very rarely switched to visit the novel feeder. So they honed in on that familiar feeder and really never left it. So that brings us to the 50-50 mix. What are those bees doing? Are they going to be more low Li? Are they going to be more high Li? What's going on? And we actually find the same, very similar trend to what we see for those high Li bees, that they are finding that familiar feeder and they are sticking to it. And even when we introduce the novel feeder, they rarely switch. So pretty interesting. So this is kind of where we were very surprised. We're like, okay, not our prediction. Come on, bees, cooperate with us. But we have a cool result no matter what. There's differences. So we wanted to dive into what's going on here with why they prefer the familiar feeder. So I mentioned that we're able to keep track of who's visiting what, which feeder, and which we keep track of the line. So in the mixed feeder, we know the high Li and the low Li in those mixed colonies, who's visiting what feeders. So then we're able to drill down into what's happening in those mixed colonies by line. So these are data just from the mixed colonies split up by the line of the bee, and number of visits split out by familiar and novel feeder. So what we find here is that in these mixed colonies, the high Li bees visit the familiar feeder, and the low and control bees are also visiting the familiar feeder. So when they're by themselves in their only low or only control colonies, they're visiting all feeders equally. But in the mixed colony, when you have the group, the high Li group, they're actually visiting the familiar feeder more. So there's this switch that happens within these lines in the mixed colony. So we're like, okay, that's interesting. What's going on? Why are they switching? So just to summarize, so this is a lot of data. I just want to take a second to summarize what's going on. So colonies with the different proportions of learning phenotypes behave differently. The high and the mixed colonies, so the 100% high, the 50-50 high-low mix, they prefer the familiar feeders. The low LI colonies visit all feeders equally, and the control colonies visit all the feeders equally, so they show no preference. In those mixed colonies, though, we see those low and control bees switch to preferring the familiar feeder. So something's going on in those mixed colonies, and we wanted to figure out what. So why? What's going on? So as I mentioned earlier, when bees are out visiting food locations, they come back and they communicate about that using the waggle dance. In these short tents, they're actually using the round dance. So this is the round dance. Sometimes you get a waggle in there every once in a while, but instead of this line of vibration, this line of waggle, you just get more of a devolved round. So the bees are circling, they're switching directions, and they're sometimes vibrating, but most of the time just performing this round dance. So we wanted to dive into what information is being shared as they're communicating about these feeders. And so we were able to set observation colonies up in the mix for the mixed LI colony. So this is the 50-50 mix. And we video recorded all the waggle dancing that was happening in the colony. So we had one frame video recording throughout the day as they're visiting these feeders. And we're doing the same thing. So we did this the next year, but we're doing the same exact assay where we're putting out these feeders, we're marking where they're going, and we're marking, we can keep track of those lines. Which lines the bees come from. Okay, so we're diving into waggle dance data. What are the bees actually communicating about? So we can split this up because we have both high, low, and controlled bees in the colony. We wanted to know who's dancing and who's following. So this is just proportion of behavior. So who's doing what in the colony. And we find that, so yeah, stack bar plot, so just showing proportions of who's dancing and who's following split up by each line. So we surprisingly find that the low LI bees are doing most of the dancing and most of the following in the colony, where the high LI bees doing equal proportions, but doing less overall, and same with the controls. So low LI bees, they're the dancers, they're the followers. They're doing a lot of the work in the colony. Then we wanted to see, when we were looking at these data, we noticed that there were some bees that were dancing and no one was really following them. So we wanted to see, how does it actually break out by line? So across the x-axis here, now I'm breaking it out by who's dancing and what line they come from. And I'm going to show you the proportions of dances followed. So what we did was just characterize, was this dance at all during the duration of the dance? Was it ever followed by another bee, or was it never followed? So it's just this like followed or not during the duration of the dance. And what we find is that the low LI bees, even though they're doing most of the dancing, they actually have a very high proportion, 27% of their dances are never followed. So they're out there dancing their little butts off and nobody cares, which is really sad for those low LI bees. The high LI bees, though, they're going viral. They're the ones that are dancing and they're garnering all the attention. So they have a significantly lower proportion of dances that were not followed. So those high LI bees, they're doing something different. They're not dancing as much, but they're garnering more attention when they do. So what's happening? So then we looked at intensity of the dance. So we wanted to, when we were characterizing who was dancing and who was following, we counted when the bees were dancing, how many times they completed a turn or switched directions. So like when they complete a turn, that's a 1. When they come back and they switch directions, that's a 1. So we wanted to characterize how vigorous they were dancing. So the vigorousness, the intensity of the dance, correlates with how the bees are perceiving the quality of the food. So we did that across the lines as the bees were dancing, and we found that sure enough, the high Li bees and the control bees are actually dancing more vigorously. So they're performing more turns per second in their dances compared to the low Li bees. So it seems like those high Li bees and the control bees, when they're visiting the feeders, they're all visiting the same feeders. But when the high Li bees are visiting the familiar feeder, they're getting more excited about that familiar feeder than the low Li bees are about the feeders that they're visiting, including the novel feeders. So, in conclusion, kind of taking all of this together, it seems like latent inhibition. So, we initially thought it'd be high Li bees are going for the novel, low Li bees won't care. And it's not actually what we see. So, there's this theory in learning, learning and cognition called conditioned attention theory. That when bees start to focus their attention, that focus allows them to ignore potential other unimportant information. So we actually think that those high Li bees are just more focused. And once they find something that's valuable, they focus in on that and value that. And so that can be when we're in the lab and we're giving them these pre-exposures, these familiarization of the odor, there's no food associated with it. But in the field, they're never going to visit an empty feeder. So we have to give them some kind of reward. And that reward will supersede every other information or all the other information that we at least measured about novelty for those high Li bees. So those high Li bees seem to be very focused, and that allows them, that focusedness allows them to focus their attention on important information. So, and that is food. For the high Li bees, seems like food is the most important thing. It doesn't matter about novelty, it just matters about food. The low Li bees, they're flexible, they're ready, they readily learn everything. So they're ready to take on information from the high Li bees and switch or do whatever is the most valuable at that time. So I think those low Li bees are great facilitators. They're the ones who are like, this is most important. This is the best one. Cool. I'll go there. Doesn't matter to me. And it's really interesting. So it seems like, again, everything's equal in the tents. There's no difference in food quality. But those high Li bees seem to be getting more excited about their food location, even though it's just as valuable as everywhere else. And the low Li bees, they're just like, meh, everything's fine, everything's great, everything's equal. So that attention to those familiar feeders from the high Li bees translates into dance enthusiasm. So anyways, that was really fun. So I published all that work in PNAS this past summer. It was really fun. Lots of time, lots of work, lots of 110, 115 degree days in Arizona, but definitely worth it. Really, really fun work. And I got to work with amazing undergrads and high school students. So really cool. So that's kind of the collective. So what's going on at the collective? Now I want to dive in for the last little bit of time into what's going on from this mechanistic perspective. So what could be dictating, what on the mechanistic level, what proximate mechanisms may be influencing this difference in cognition and therefore foraging behavior that we see? So first I wanted to dive into kind of neurophysiology. So I have a little bit of experience in that from my PhD. And so I wanted to dive into what neurotransmitters were going on in the brains of these bees as they were doing different behaviors. And then we also dove into some electrophysiology, which Brian Smith's lab is they're experts in. So when we were collecting scouts and recruits for that first study, so the individual level behavior, when we were collecting scouts and recruits to test them for their LI behavior, we are also collecting them to analyze their brains for different neurotransmitters. So we're able to use this technique called high pressure liquid chromatography, or HPLC. I'm sure many of you are familiar with this. And we're able to assess the amounts of octopamine, dopamine, tyramine, and serotonin in the brain tissue of these bees as they were foraging. So as soon as they got back to the colony, we grabbed them, threw them into liquid nitrogen, dissect out their brains, pass it through the HPLC and characterize the amounts of neurotransmitters. So here I'm just showing you the results of tyramine. So tyramine was the only big difference we saw in scouts and recruits, and we actually find that the scout bees had slightly higher amounts of tyramine in their brains compared to the recruited bees. So this work, a lot of this work is published in was published in 2018. So if you want to dive into those details, please feel free. So we see differences in neurotransmitters. That could be a pretty interesting place to go to. Then we wanted to look at what's going on in the antennal lobe of the bees when they're receiving this information. So we're able to, in honeybee brains, we're able to dissect their heads. And while they're still alive, we can insert a probe and do extracellular electrophysiology, where we're just kind of characterizing the global responses in the antennal lobe as bees are experiencing different odors. So we have this probe. It's a little, I mean, it's hard to not be able to point, but if you can, I hope you can see my mouse. Cool. So you see this probe. It's a two-pronged probe that can be inserted into the antennal lobe, and we get these waveforms as the neurons are firing that can characterize intensity of the responses, the general intensity of responses in that tissue in the antennal lobe. So we get these spikes from all of these different locations on these probes. And then we split the odors that we're giving them out into novel and familiar, just like the assay that I showed you in the beginning. So we give them, we pre-expose them to two odors just to make sure there's no difference in these two odors. They're hexanol and octanone. So we puff odors at them. We assess their the antennal lobe responses to those odors, and then we pre-expose them. So we puff odors at them. We pick one, we puff that odor at them 40 times over 5 minutes, so 3.5 hours. So the bees are in this rig. They have the probe in their antennal lobe. They're getting this exposure. We wait 10 minutes, and then we give them a post-exposure. So how does the intensity change? The intensity of those responses change to the novel odor. So now this is a novel odor versus the familiar odor that they just were exposed to 40 times. So just to shout out Hong, who's a research professor at ASU. He is an electrophysiology extraordinaire. He's fantastic. So he really spearheaded this work. And an undergrad, Eric, who is also just absolutely incredible in this work. So I'm going to show you results. So this is the post-test minus the pre-test. So most of these values that I'm going to show you are negative. So we see a decrease in response over time, but the more negative, the more the fewer neuron responses we're detecting. So so I'll show you first the results for novel, and then the results for the familiar odors split out by the scouts and recruits as we collected them. So what we see is that when you test, so the post minus the pre, so what's happening before and after to that novel odor, the odor that was not pre-exposed, no difference in responses in their antennal lobe to that odor. So the recruits, the scouts and recruits, they have a decrease overall, probably because they've been in that rig for 3.5 hours, or probably closer to 4 hours at this point, but no difference between scouts and recruits. However, when we look at the familiar odor, so this is the familiarized odor that they've been pre-exposed to, we see a dramatic drop in the scout bees' antennal lobe neuron responses. So overall, those scout bees actually show a reduced response to the familiar odor after they've been exposed. They've both been exposed the same number of times. So I think this also continues to kind of touch on the mechanism of potentially habituation, or the learned response, or what's happening kind of in the proximate tissue. So this is way before that this information gets processed at all in the brain. This is really in the proximate, just like sensory tissue in the bees. So we see in those scout bees, so scout bees, again, they're more high LI, they ignore the familiar odor in their behavior. So it seems like there's something going on in the antennal lobe that's mediating that ability to ignore that odor. So pretty interesting stuff. Hong and I are working on publishing these results right now. And now I want to dive. I know I'm getting towards the end, so I want to leave time for questions, but we have so much cool stuff and I'm really excited about it. So really quick on the transcriptomic stuff. So I wanted to, part of my NIH postdoc was trying to identify some of the gene regulation and the gene pathways that may be influencing cognition, the high LI, low LI, the scouting and recruiting behavior. So I designed this kind of ridiculous before I knew about RNA-seq nested experiment where I really wanted to dive into kind of parsing out what was the main driver to differences in gene expression in the brains of these bees. Is it the genetic background, so the selection that we did on them? Is it whether they're scouts or recruits? Or is it whether they're high or low after we've tested them and assessed their learning behavior in the lab? So what we did was we selected them. So we have these lines and we performed this hive moving assay where we collect them as scouts or recruits. So we did that to each of those lines. So we forced them to be scouts or recruits and collected them. And then, so we collected them at that stage. And then we had a subset that we brought into the lab and assessed their learning behavior. So we have frozen bees that are scouts and recruits just returning to the lab. We collect them at that moment in liquid nitrogen, and then we take a subset into the lab. We assess their learning behavior. As soon as we're done with that LI assay, they go into liquid nitrogen as well. So I have 105 samples of all of these combinations, and I want to know what's driving the gene expression and the gene networks that may be influencing these behaviors. So first I'm going to show you the results, the very broad results from these control bees. So these scouts and recruits, so you can see the colors along the top, the pink and blue along the top. These are just separated out in space based on gene expression, so differential gene expression. And they separate out beautifully by scout and recruit based on differential gene expression. So these are control bees, so they're not selected. So we just assess their differential gene expression. They separate out beautifully. So that was really cool. We're like, okay, there are some genes. There's an odor receptor in the brain that seems to be playing a role, differentially expressed gene. Pretty interesting. Then now I want to show you the scouts and recruits from the selected lines. And so this is the same heat map of differentially expressed genes. Now we've layered line on top of this. So this is the selected low or high line that we selected the queens and drones and we're getting workers from. And you can see here that with that selection, that beautiful like arrangement of scouts and recruits totally disappears. So we know that when we select these bees, it changes kind of the longer term, the gene expression that can happen in the brains of these bees. So really interesting. This is literally just scratching the surface of the iceberg here. Like we have a lot of stuff, 105 samples. I'm only showing you 50 right here. So we have a lot of data that we're filtering through. We're getting close, but hopefully this work will be out this year and we'll be able to give you some more definitive answers on gene expression and gene networks and cognition and foraging behavior. All right, so with that, taking this multi-level approach, I love being able to dive into this collective behavior, looking at the individuals, looking at the mechanisms, and I'm very thankful that throughout my career, I've been able to get a lot of experience with things like being out in the field and doing these colony level behaviors, but also being in the lab and being able to dissect brains and do RNA-seq. So a lot of these skills serve me well to be able to set up a lab, and I'm really excited to continue this work at Marquette as a new professor. So as slow as things move when you're a new professor, Brock, I'm sure you know this well, it's definitely interesting, especially during COVID, but I'm really excited, really stoked. So yeah, collective behaviors are really awesome. I think that it's really important to think about collective behaviors holistically, and that's the goal of our lab. So with that, I know I'm like pushing time, maybe have a little bit of time for some questions, but if I don't get to your question or you want to chat more, please get in touch with me, email me, tweet me, whatever works for you. And thanks so much for your time.
That was really cool. Thank you. I love the holistic approach.
It's fun.
So we do have time for probably one or two questions, and I see there's one from Matt Dittman in the Q&A right now. And Matt is asking, Since high/mixed LIBs seem to prefer familiar over novel food sources, all else being equal, how quickly do you think they'd switch to the more novel food source if it were found to be of higher quality? So you can read that there if you need it to.
Yes, I see it. That is such a great question, Matthew. So we have a paper in review right now about this exact thing. These data that we have that hopefully will be out soon show that, and we only did this with high Li colonies and low Li colonies. We did not do mixed colonies, but those high Li bees don't switch. So even if we give them, we offer a feeder that is, we doubled the quality. So I think we had 1 molar and we had 2 molar novel feeders that we put out. Those high Li bees do not switch. The low Li bees, they switch they're like ready to go, ready to switch. They switch very quickly. The high Li bees don't seem to switch. So another really interesting, so again, like how that doesn't make sense. It's better quality. Why aren't you switching? But I think this again kind of gets to that, just like maybe almost like detrimental attention. They're too focused on those, the feeder that they find like initially, and that's familiar and reliable. And that's what matters more to them than novelty, and even if novelty and quality. So we still have to, I think that there's a lot of room to do almost like a dose response curve. Like how much better does this need to be to get them to switch? So we haven't gotten there yet, but that's a really great question, Matthew. So hopefully that data will be coming out, will be published soon, and you can see some of the weird results that we get. Great question.
Awesome. Matthew says, neat, thanks. We can probably take one more. And then Dr. Cameron sent a message here. He said, it's okay if you want to take a few more and be a little late to the meeting afterwards. So
okay,
we'll take one more, see if any more roll in. And so Crispin is asking, what environmental factors impact LI foraging? So are there any environmental impacts?
Yeah, so I think Matthew's question kind of touches on difference in food quality. So I think that is a major environmental factor. So like what's available, in the environment, what are bees experiencing, what's their the history. So I think that there's a lot of environmental factors just about the food quality or the food availability and location that may influence it, influence LI and differences in foraging. And that's, the really interesting thing, again, this kind of like this multi-level approach, we were thinking about, how could LI even this variation come about? Is it strictly genetic? And if it is, in a typical honeybee colony, when there's many, like 12, 15 different patrolines, how is LI potentially being like dictated in the colony? Is it genetic? Like is the queen laying certain patrolines because of their high and low LI because of environmental factors, or is there some environmental factor that influences and drives the phenotype to high or to low? And the cool thing about that is that at the colony level, there's no difference in that. It doesn't matter, right? You're still going to get the same result at the colony level, meaning like you're still going to get high or low bees regardless of whether it's genetic or environmental. But what actually could potentially drive it if it were environmental? I would say like food quality coming in, amount of food coming in, and then potentially what's going on with the foragers. If there are, if it's equally distributed among, across the environment, or if you have patches, right? So where if you have an environment that's more patchy, and I think this is like Anna Dornhaus has done some of this work where the patchy environments are the environments where the waggle dance really matters. And so they're actually not wasting a bunch of energy exploring, and they're actually coming back, and the waggle dance is super valuable in these really patchy environments. So I think food distribution of resources is probably one of the biggest environmental factors that may dictate. So if you have a patchy environment where bees find this food source and they are really excited about it and drive everyone to that one food source, instead of bees that are kind of just like lollygagging around and like whatever, everything's equal, and saying, nope, if you waste your time, it's going to be detrimental to the colony. Go to this colony. And those low bees are like, sure, let's go. That's fine. Cool. Let's go. And they just all go there. Then that could be advantageous, right? So there's a lot of, it's a super complex issue. And I think that that's such a good question to think about, like, what's the environmental factors, and the selective pressures really is what that question is also getting at, of driving this variation. So really, really cool question. Lots of directions for sure to go in.
Yeah. I'd imagine that pesticides also impact a little bit too.
Oh my God. Yeah, absolutely. Right. I mean, we know, even just on a cognitive perspective, that pesticides change like cognition in bees and how they can learn and remember. So like if a high bee is like really excited about a place and then like experience a pesticide that changes that could really influence like how they waggle, how they recruit, right? So yeah.
So I think we've just got one more question here. It's a super quick one. How much how much mortality do you get in 2 weeks in a high tunnel experiment?
Oh, great question. So we, because we keep the bees inside and do not let them forage on, whatever's in the environment. They only, they're excited about what we give them in the tent. So we started this with colonies that we just let freely forage and then put in the tent, and we had just like massive death, right? Like if they know that there's better stuff on the outside, they're like, this is horrible. I don't want any of these things. And they will like try to escape so hard and kill themselves. So that's what we figured out, leaving them inside for 2 weeks and just like spritzing them with sugar water, giving them a frame of honey and a frame of pollen that we remove as we put them in to kind of motivate them. Then we don't see as much death, which is like what that was our solution. So we actually don't see a lot. We didn't do a before and after, but just kind of anecdotally, like seeing what's going on in the tents, we didn't see as much death by doing that. Great question, because that is a major hurdle in doing these flight cage experiments.
Okay. Well, thank you so much for your time today.
Thank you. This is so much fun.
And thanks everyone for sticking around to the end here. Grad students, if you have questions for Dr. Cook, you can also, and please do go to the grad student lunch at 12:30 Eastern, 11:30 for you, Dr. Cook.
Yep.
And great, thanks everyone.
Awesome.
Enjoy the rest of your day. See ya.
Bye.